So I think it's 12. We can probably start. I'm very happy that we have here Lisa Pokorny and Paul Fernández Mató with us for our June seminar, ERGA seminar on biogenome analysis and application seminars. We'll have two talks by the speakers. So I will start by introducing the speakers. First one is Lisa Pokorny. She's a Ramón y Cajal researcher at the Real Jardín Botánico, Royal Botanical Garden in Madrid. She studied in the Autonomous University Madrid and got a PhD from Duke University in the USA. And then she had a postdoctoral experience in several institutes, such as the Botanical Garden in Madrid, the Kew Botanical Gardens in the UK, the Center for Biochemistry and Genomics of Plants in Madrid, and the Botànic Institute in Barcelona. Her research focuses on understanding the biodiversity, generally speaking, and she is known for implementing several high-throughput sequencing approaches, namely the target capture sequencing, which she will talk today, and several very well-known bioinformatic workflows. And after Lisa, we'll have a talk by Paul Fernández Mató. He's a PhD student at the Institut Botànic de Barcelona. He graduated in biology from the Universitat de Barcelona and did a prestigious Erasmus Mundus program, Tropimundo, thanks to some La Caixa fellowship. And he's currently, I think, completing his PhD thesis under the supervision of Jaume Pellicer on the evolution of giant genomes in plants. Very exciting topics. So with that, I will give the stage to Lisa. Thank you very much.
Thank you very much. Let me share my screen for that introduction. Iker, very kind of you. It is a pleasure to be here today. And without further ado, I will start talking about target capture sequencing approaches and their applications for the study of plant evolution. So I have a structure, my talk, a little bit like a very short intro, why plants, what's the deal with their genomes, what is this HypeSec that we use so much in plant systematics, and how you go about designing kits for target capture, how you implement both a molecular and dry lab workflows, and then things you can do with them. And Paul will give us a more in detail example of the cool things you can achieve with target capture sequencing. So what's the deal with plants? Well, they're autotrophs. Very few organisms or lineages out there are autotrophs. Then they have this crazy life cycle where they have alternation of generations. So you can have haploid and diploid individuals, which can be or not independent. Then they have a very long history of evolution, more than 1,400 million years of history for the entire plant kingdom. Although most folks know of land plants and more particularly flowering plants. But yeah, it's a very large lineage that achieved the transition to land from sea, which in itself it's one of the large transitions. So what's the deal with plant genomes? I'm going to talk a little bit about the genome architecture and their genomic dynamism. So turns out genome size, it's sort of a spectrum of possibilities. And in angiosperms, it can be like 2,450 times range span. But in ferns, we recently, and this is Paul's PhD work, measured 160.45 gigabase per genome in this tiny fern. This thing's small, okay? This is a very, very close up. And these white dots you see here, Those are the stones, because it has huge cells to store all of that DNA. But as you can see, most plants have on average much larger genomes than animals, with a few notable exceptions, or fungi. Okay, so that complicates sequencing their genome. So why are they so big? They have lots and lots of repeated elements. They actually were described by Barbara McClintock on Maze, and she got the Nobel Prize for that. So yeah, repeated elements abound. Then they do polyploidy. They do whole genome duplication. Their meiosis sometimes doesn't work. The gametes are unreduced. They join together, and you end up with an individual that instead of diploid is tetraploid. And this is sort of like a cyclical process where you end up with a whole genome duplication, and then there is gene loss and genome stabilization, and you become diploid again, and on, although sometimes you go very many times polyploid. What happens as well, and there's a lot of hybridization and subsequent integration. So species boundaries are sort of porous, and then sometimes they just go crazy because their genomes are not stable, and they sort of go, okay, vegetative reproduction, I'm going to be clonal because that's the only way I can survive with this mess of a genome. And it's complicated, and it's reticulated, not bifurcated. So plant genomes are awesome. So what do you do when you have such an interesting and fascinating system that has lots of, let's say, exceptions to what most people are used to? So you can try this approach that we call HypeSec, following a paper by Whitemeyer and colleagues published in 2014. Where they combine target capture sequencing with genome skimming, or also called nowadays low coverage whole genome sequencing. So when you do low coverage whole genome sequencing, you're going for anonymous regions in the DNA. What you're going to get is whatever it's in highest copy or higher copy number. So you can get organellar DNA. It's going to be good to get yourself some plastomes or mitomes or whatnot. But as I said, it's not really target. You may even describe the repeatome, the repeated fraction that we were mentioning earlier. But if you want to build phylogenies, understand relationship, you need orthologs. And the really neat thing of target capture sequencing is that you can actually, if you have some prior information, okay, this does not come from the void. You have some prior information, you can design a kit to retrieve your orthologs through this in solution hybridization with biotinylated probes, which you can later drag with these magnetic beads that have a coating of a streptavidin. So the streptavidin and the biotin sort of join, and you get your DNA, and magic, you get all your single copy genes that were like in very, very, very low abundance. And that's really neat. So if you combine the two, then you can have whatever is in high copy with all the genes that you need for phylogenetic reconstruction. So I always put this funny drawing slide. So when we do the Sanger sequencing, we sort of have a bait, and that bait is going to get us a fish. When we design a probe set for target capture sequencing, sort of have a net, and we're dragging it, and we're getting all of our single copy genes that we have boosted, and then we're going to get a lot of things of target that are there because we're doing this. We're combining with the low cover whole genome sequencing. So we're going to get a lot of information. But how do you go about designing these probe sets or bait sets for target capture sequencing? So, I mean, like everything in life, you need human, genomic, computational, and economic resources. So you need folks that can actually do the wet lab. folks that can analyze the data. And then you need, as I said, prior knowledge. So this is the 1000 plant transcriptome project, or consortium initiative. It ran from 2009 through 2019, which is when we published our capstone. And we ended up with the largest phylogeny for all of Archaeoplastida. So this includes 410 orthologs shared across Archaeoplastida, so plant kingdom. And interestingly, here in this bit here in this corner here, we have 600 angiosperm flowering plant genomes and 20 or so genomes that were available at the time through Phytozone, which is the Joint Genome Institute web for plant genomes. Of course, nowadays, more and more, you can have genomes. Indeed, we continued our 1000 plant transcriptome initiative through this Open Green Genomes initiative, where we've generated 35 additional plant genomes that are widespread, phylogenetic widespread, to cover gaps of knowledge. But there is also this Chinese initiative, 10,000 plant transcriptome. ERGA is contributing. It's part all of this of the Earth BioGenome project. And then I have to say Darwin Tree of Life is the one that has contributed most plant genomes, in addition to Open Green Genomes have as of late. 10KP, they're producing lots and lots of data, but most of the data is being processed for blastomes, and I really want to see the annotated genomes start to come. In any case, you have all of this information. It's huge, and you have to process it. So high performance computing is the way to go. These are two supercomputing clusters that we have in Spain, the Barcelona Supercomputing Center and the Galician Supercomputing Center, which are really nice, super nice stuff that help us out. Well, and then here I'm gonna give you an example of a kit that we designed back in 2017 and finally published in 2019. So this is a collaborative effort with my colleagues, Matt and Norm, who are now in Texas Tech University and University of Vienna. And then here we have Ilya Lich, Felix Forrest, and Bill Baker. They three were leaders in different capacities of the PAFTL project, which is sort of where the framework where we designed this kit. Ilya is now very involved in Darwin Tree of Life project as well. So for your information, flowering plants have around 370,000 species total. The Angiosperm phylogeny group on its fourth iteration recognizes 416 families and 64 orders, or grouped in, and they have importance. I mean, they are part of our everyday life. So as I said, we use these 1kp transcriptomes and then 20 genomes that were available, and then we did some mathematical there. Calculated timing distances, used k-medoids clustering, and we joined forces with Dysel Arbor Biosciences who are based in the States to design a kit that targeted 353 nuclear orthologs with a data matrix of this size and probes that had 3 times tiling. So the idea with this, now you have a tool that works across angiosperms. How do you go about implementing it? And what can you do with it? So wet lab workflow, I had a bunch of longer slides, but I've sort of condensed it. You can extract your DNA. The advantage here is you don't need high molecular weight DNA extractions. I mean, these are not genes, you don't need RNA. You can work with a 200-year-old herbarium collection it will work just fine. Actually, one of the earliest steps once you extract your DNA is to chop it, okay? Then you prepare your genomic libraries, usually dual indexing, so that you can multiplex. And I think I have to go faster because I'm going over time. And then you enrich with this probe set that you've designed and sequence with your high throughput sequencing platform of choice. You can do short reads or long reads depending on how much you have chopped your DNA in early steps. We shared a bunch of our strategies in this paper early on, so please do check it out. Then the dry lab pipeline. So we're going from raw reads to target sequencing to those target sequences we're interested in. So I like to describe this bit like a cut copy paste color bit, where you have clean your reads and then assemble target sequences. Again, my colleagues Matt and Norm created this amazing workflow pipeline that I've been using for many years now, and it just gets better with time. So now you have your target sequences. You need to assemble your data matrices and get alignments so you can do phylogenetic inference. So this is sort of like let's say a washer, when you're washing clothes, all the cycles that you have, first you wash, then you rinse, and then you repeat a few times until you get everything in place. So you try aligning, then you improve that alignment, you remove like missing data and optimize informative data, or you remove outliers from your data matrices. Eventually you have your alignments, and now you want to have your species trees, gene trees. So this is like a little bit of masonry. You have your bricks that are your gene trees, and then you need a little bit of mortar so that you can end up with this gorgeous wonder wall. So as I said, the number of tools, maximum likelihood is a go-to approach. A few in between tools that can help you polish and work with these gene trees, and eventually you can build not only species trees, but as I said, plants, reticulates, so networks. And then super fast now, now we have our kit of angiosperms. We published last year in Nature this phylogeny of angiosperms that includes 60% of all known genera of angiosperms. We've revised the classification of angiosperm compared to these mostly plastid markers, and there's a lot of incongruence, which is very interesting. But another cool thing that we can do is have talked about phylogenetic sort of workflow. You can, with the same data set, you can use a population genomics or population genetics approach, and basically you can extract SNPs. And once you extract SNPs, you can do very cool studies where you integrate recent collections with herbarium historical collections from 200 or however many years old, and you can understand the phylogeography, for instance, of both wild growing and cultivated cannabis. We just published this out. I hope you like it. Or this one that we have in review in collaboration with Regina Berjano from University of Sevilla, where we've been able to show that there is gene flow between ploidy levels, so diploid, tetraploid, in a polyploid complex. So they are not instantly or completely isolated. As I said, plant genomes are porous. So I hope our reviewers and editors like it, and it's soon out. And without further ado, thank you very much, and sorry for stealing so much time from your talk, Paul. Thank you.
Hello, everyone. I'm Paul, and I'm gonna present. So what Lisa just explained you is the the whole theory about it, which is a lot, and she did very well to summarize it all in 20 minutes. Now I'm gonna present a case study that will likely and hopefully show you the advantages of target sequencing and what can you do with it. And especially in a case study that you're going to see, it's very unique and difficult to work with because we are talking about giant genomes. So let's start by presenting you the group. This is a group of ferns that is called Mesipteris. It's a genera. It consists of 17 species before this study. They are mostly epiphytic plants. They live in human mountain forests. You can see that their distribution is in Australia, New Zealand, New Caledonia, several Pacific islands, also Indonesia. And some of them, and this is important, are in very remote and inaccessible locations that we could not go to sample because of COVID, political instabilities, or weather, for example. Now I'm going to show you some pictures. These are tiny ferns, as Lisa said before. They are mostly epiphytic. You're going to see that most of them are epiphytic, but there are some that grow from fallen trees and bark, and they have this erect kind of growing, like Mesipteris articola or oblanceolata. And you may be asking, but why don't we base our studies in whole genomes? I just did some numbers a couple of days ago, and in NCBI, I found that just 11 species have reference genomes for ferns right now, versus, for example, 2,200 in flowering plants, which is still low, but in ferns is dramatically low. And why is that? So there are many answers to the question, but two of the main ones is that ferns have high ploidy levels and chromosome numbers, which makes for difficult and costly assemblies. And they also have big genomes. So let's go a little bit deeper into that. Ferns have a mean chromosome number of 137 chromosomes and a world record of 1,440 in the species with the most. In our case study in Mesipteris, we find two different ploidy levels. There are tetraploids and octoploids. Here you can see in the left an image of one of our samples, and in the right you can see chromosome counts by other scientists, Perry et al., in which you can see the difference between a tetraploid and an octoploid. As you can see, the chromosomes are not especially small because these plants have very large genomes. The genome size in plants, Lisa summarized it very well, it varies a lot. In ferns specifically, we find a range of 208 times difference between the smallest genome, which is 0.77 gigabases per 1C, and the largest one, which I'm going to talk about later. But the mean genome size is rather big. It's 14.61 gigabases per 1C. And in our group, we find the largest genome ever. As Lisa told you before, the group genome size goes from around 70 gigabases to 160 gigabases. So with these very large genomes, the only option we have is not to sequence the whole genome because it would be impossible nowadays. It would require a lot of resources and time. So what we did is target sequencing. Now I'm going to go through the materials and methods, but I'm going to focus on the target sequencing part because it's the base of the study, but I'm going to show other results. If you have any questions about any of the other methodology, please ask me after or contact me after also. We have a lot of samples. Most are from herbarium. This is going to be important for afterwards. But we also have some fresh samples from our field trips. in New Zealand and New Caledonia. The target sequencing design was to make a custom kit. We mined transcriptomes of Xylotum nudum, Mesipteris parva, Mesipteris thanensis, which were the ones that were available at the time for low copy genes, described in 1 kp, resulting in 288 target genes. These were highly specific targets because they were extracted from the species we were interested in studying, and we had 3 copies per gene, 1 for each of the species. We are trying here to increase success rate because we are talking about giant genomes, and if you imagine a bait of 120 nucleotides, which is what we designed, trying to find a gene in 160 base pairs, it's different than trying to find a gene in a genome of Arabidopsis, for example, which is very, very, very small. So we were trying to increase the success rate as much as possible, and that's why we designed this kit, very specific, with 3x styling, with approximately 17K baits. So the results from the target sequencing. First, the numbers. We got from the 288 genes, 260 were without paradox, so that is a very nice success rate. Bear in mind that these are polyploids we are talking about, so they maintain still these genes in very low copy. And in terms of samples, we had good recovery data for 70 samples. What happened with those? So the fresh ones worked fantastically. We had 100% sample success, meaning that target sequencing works in giant genomes just fine. But the problem we had is that this sampling from for fresh samples was limited, and we did not have all species. So what the herbarium samples allowed us was to complete the sampling from places we could not go nowadays. And we also were able to recover genes from samples as old as 1862. With all this, we built a nuclear phylogeny that is a well-resolved dated phylogeny of all species known, and it was divided in 2 clades. I'm gonna use the pointer here. In the Pacific clade and the Tasman clade. We also saw that there was phylogenetic signal for a life form. Do you remember that I told you before that some of the species grow erect and not epiphytic? So they are in a subclade inside the Tasman clade. With this phylogeny, we are also able to plot the genome size and the ploidy levels of these species to find at least 3 whole genome duplication events in the phylogeny, with potentially another one that we will not talk about because these are new species. Especially, we found 3 new candidate species, ones that did not match any of the other clades. And we also found a clade in which the supports were not great, and the distances between the species were very small. And we found several described species, so we would think that a taxonomical revision is needed, especially for this group, since all the other ones were well defined as species. And finally, we found that most diversification events occurred between 30 million years ago and 12 million years ago, which coincides pretty well with the reemergence of New Zealand that was submerged and other geological events. So we were curious and we wanted to know the diversification history, and with a well-resolved phylogeny at the species level, we could run BioGeoVerse in R to find that most diversification happened from New Caledonia and closed islands, and some from Australia to all other regions. Until now, what I've described, these results are part of the direct results from the target sequencing data, but we also complemented this analysis with other analyses, especially from genome skimming. As Lisa told you before, you can do HiveSec, which is doing target sequencing, but including a little bit of uncaptured library, a little bit of genome skimming inside the same sequencing process. But we didn't do that because, as I told you, we are working with giant genomes and we wanted to maximize the success as much as possible. So we did it separately. But in case you do hybrid sequencing, you could do this with the same data. What we did here is a plastid phylogeny with all species again that has 60 plastid genes. What we found here, comparing it with the nuclear one, are incongruences that are pointing at possible hybridization events. We also find some clades, for example, the one here that was problematic, that is also very mixed up when we look at the blast. And another case, which is this clade here of the erect growing plants, most of them that are mixed up, but this is probably due to recent diversification and that the plasts are not different enough to give us a well-resolved plastid phylogeny, as you can see also in the supports. And finally, Lisa also talked about the genome composition and the repetitive elements. And since we are working with giant genomes, we are very interested why they are giant genomes and how do they arise. So we are interested in the genome composition. So we were able to analyze several species. Here I just gave you a subsample in which we find the largest genome of all of Lanceolata, since we were very interested in knowing how such a big genome has formed. Before, I already told you about the whole genome multiplication events that happened, but we also saw that there is an accumulation of all kinds of repetitive elements in this genome compared to the sister ones. There is an amplification of all these kinds of repeats, and especially satellites. Just to mention that this analysis was done using 3% of the whole genome. And this took weeks for computing, weeks of computing for each individual. So this puts a little bit into perspective the advantage of target sequencing, which allows us to work in giant genomes almost as we do in very small genomes. While one where we are talking about genome composition or whole genome sequencing and assemblies, we are talking about an exponential increase of resources and time. So finally, I wanted to give some takeaway messages. The first one is that we have an efficient gene recovery for phylogenomics using target sequencing in the largest known genomes, with some moderate success in herbarium species, but still enough to have a whole and complete sampling. So the first takeaway message is that target sequencing works for giant genomes, and especially if they are fresh, it works perfectly. There is potential follow-up analysis depending on initial data sets. Lisa already talked about the population genetic analysis, but you can also do some biogeography. There's combined with genome skimming allows to tackle issues such as hybridization or genome composition from an evolutionary perspective, which is very useful if you want to study, for example, the evolution of genomes and the evolution of genome size, which is our case, and ploidy. And is one of the fewest options feasible nowadays to study groups with such challenging characteristics as the one we are working with in our group. So with all said, I just wanted to acknowledge Jaume Pellicer, which is the group leader, and Lisa, which helped a lot with this analysis that I presented today, apart from all the people involved in the project from one side or another. And thank you very much.
Thanks a lot, Lisa and Paul, for these two very, very interesting talks. I have to say it was amazing. I learned a lot of new things. So we have a couple of questions from the audience. Robert Waterhouse asked, how much does the choice of markers for phylogenomics and the pre-processing of the data and trees influence the robustness of the resulting phylogenies?
Actually, this is one of the really cool things of target capture sequencing. I mean, I've talked about orthologs, but if you are interested in a metabolic route or pathway and you want to see if a pathway is present in a group or another, you can target the genes, the minimal set of genes required for that metabolic pathway to work. Or you can have a, let's say, mixed kit where you include orthologs for phylogenetic reconstruction. And some, for instance, genes of agronomic interest that have to do with, I don't know, like bark production or tubers or whatever. So, as I said, in order to design the kits, you need prior information. You don't do it from not knowing things. So, indeed, your choice of markers does influence what you're going to get, but let's say that the kit you're going to design or use depends on the question that you want to answer. So if your question is purely, I want to resolve relationships in this group, you go for certain type of designs, some of them already available, like our Angiosperms 353 kit, or you do something custom. And if you are interested in something more like physiology or the evolution of a cluster of orthologous genes or whatnot, then you take a different design approach. One of the really neat things also is that saves you time, because you do a lot of pre-processing before designing the kit. There is a lot less analytical steps that, if you're working, for instance, with transcriptomes, you spend a lot of time identifying your orthologs and making sure they are the orthologs you want to be working with. And by the time you're doing target capture, someone else has already done that work, or you've done it yourself. And once you have the kit, you are very familiar with the behavior of those genes. So you just go and jump into the analysis and get cool things. I mean, really nice results like what Paul was showing us. I hope that answers.
And I think that also if you are targeting species phylogenies, in principle, choosing one set of markers or the other, given that the vets have been decided to identify orthologs that are supposed to reflect these species phylogenies, it should not matter too much because the underlying phylogeny should be there unless we have very complicated cases, no?
I mean, yeah, reticulation happens.
Okay, the next question is by Camila Mazzoni. She asked, how exactly do you define that you have caught or not paralogs?
Well, we used a very conservative approach, meaning that if there were more than 2 copies, even though they were polyploids in our case, but we use the same for different. If there are more than 2 copies of a certain gene, we consider it that it has paralogs. I want to mention that there are tools nowadays to try and analyze those paralogs and include information in the phylogeny, which is something I haven't explored in detail, but I'm aware and I think it has a lot of potential. But we just use the ones that had 2 or fewer copies, 2 or 1, basically.
Yeah, these sort of stems of the pipeline that I was explaining, my colleagues design, HiPiper. So HiPiper is really neat. Not only does it assemble your targets, it gets you very many statistics to really understand your data set. And one of the things that it calculates is like whatever target you have assembled, in how many copies does it appear given the data, the input data, and the reference, which is the file with the targets that you design, you used to design the probes, which you can bioinformatically modify so that it is more accurate and whatnot. But the really neat thing is that this pipeline tells you, sure, you have 2 or more, or you have 3 or more. And that helps, again, as I said, because you've chosen these genes because you know they're very likely to be in single copy across organisms that do polyploidy left and right. So we were going for 288 and we ended up with 250. I think that's a very nice ratio of single copy versus more than, yeah.
So HiPiper gives you information about the number of copies on the reference species or also on the ones that you sequence?
On the ones
so
you sequence. On the what you have sequenced.
Mm-hmm.
Yeah.
Very interesting. We have a third question by, forgive me if I pronounce it incorrectly, Guts de Seelingier. Maybe I missed that part. What is the average coverage of a genome skimming that you presented?
When I sequence genome skimming separately, not as HiPSec, but separately, we try to sequence 10% of the genome. But since we are working with giant genomes, we use less, just in case something is wrong. But the results I presented were with 3% of coverage.
I mean, if you translate that into gigabases,
yeah, that's a lot of.
It's a lot of butterfly genomes. Let's say that way. Just 3% of one fern.
Yes.
Okay, so while we wait for more questions, we have a new question from Tom Brown. I had some questions. I leave them for later. So Tom Brown asked, given the differences that you showed between the plastic and the genomic trees, as well as the hybridization that you spoke about, does it make sense to think about evolution in this way in such species? Do we need more dimensions to the trees or another way of visualizing these relationship histories?
So I mentioned very briefly in my talk and fast. So incongruence and conflict abounds in plants. Gene trees within the species trees will have different coalescent histories. And then plastomes are maternally inherited for the most part. And they're telling you a different story than the species tree story. So it is expected that you're gonna see this conflict. We usually do a tanglegram where you have the nuclear versus the plastic topology and you see how things are crisscrossing. But yeah, phylogenetic networks are up and coming, but they're very limited in the amount of tips and genes they can deal with. So more often than not, what we do are subsets of our huge data sets so that we can test particular hypotheses. I hope that answers. Do you want to add something, Paul?
No, not really.
Okay.
Great, thanks a lot. So now, one of my questions, I was curious if you made some direct comparison with the same data set of target capture versus genome screening in terms of cost of sequencing or depth per base pair that you get for those markers that you are interested?
Yeah, so we've done that for certain groups, and we've compared how much depth of sequencing you need to get to those single copy genes. Of course, it's completely dependent on the size of the genome. The smaller the genome, the more comparable is the pricing. Again, target capture sequencing is cheaper, especially if you implement all of those tricks that we share in a paper back in the day, than what you have to spend. Surprisingly, in spite of the fact that you have to do genomic libraries and pull and then get your kit, blah, blah, in order to get to those single copy, to those orthologs, you really need to go very, very in depth for the. So sometimes I actually recommend it. I've designed kits, target capture kits for lineages that have tiny genomes, such as fungi, where you could go whole genome sequencing for the whole thing. But when you're working with suboptimal DNA template, for instance, fungarium collections, and I think this is another really neat thing about target capture sequencing, is that you can integrate natural history collections into your research. So we were able to get almost 200 single copy genes for a group of 2,000 species. Cortinariaceae is a group. They're mostly arbuscular mycorrhiza, so you don't have fruiting bodies, you just have hyphae, and you don't know what that is. So it's really neat because you can work with tissue that has no morphological character, so you can work with herbarium collection that, you know, like you look at it and it's like, do you have DNA? But yeah, it does. And you can get those genes and build, understand the evolution and biology of the group.
Yes.
Okay,
I just
thanks.
wanted to add that, of course, we haven't tried with our giant genome because it's too costly to do something like that. And regarding the collections, it's very important that I presented that just 17% worked for our target species, but our target species is very special, and there's many studies that have worked with target sequencing and collections that with normal sized genomes that work much better. Just that we are trying to adapt complications here, and it's
it is an extreme example. Yeah, which is why we wanted to share it with you.
Actually, I had one question for Paul related to this. I think you commented that you design new baits in a way that they are more efficient for large genomes, but I didn't quite get exactly what the differences were with the baits that Lisa was presenting. Can you comment a bit on this?
Yes, the main fact is that the genes that we used were directly from the genus that we were studying. For example, Angiosperms 353 does some math and does some sequence that is not any plant, but it's similar to all plant or angiosperms. But here we were using specifically the genes that were found in these species that we had access to, that were these three. So that is the main design.
Yeah, so his approach is what we call a custom approach, and the Angiosperms 353 kit is what we call a universal approach. One of the really neat things that I didn't have the time to say is that the baits can be very divergent from your target, but the less divergent they are from your target, the better the capture works, of course. So the Angiosperms 353 kit is trying to get at 127 million years of flowering plant evolution, and we didn't have representatives of every lineage out there, also because it would have made for a very pricey kit. So we we use the summing distance and k-midoi clustering to identify representative sequences that were no more than 30% divergence from the other sequences they clustered with. And I mean, it's really interesting that it does work in spite of the fact that some of the things that we're using these baits for are 30% divergent from the baits we're incubating them with, and you can still get the data and process it. So that's really neat. But as Paul was saying, when you have something more particular, when you have a very, I mean, when you have a group that has gigantic genomes and is polyploid and has lots of repeated elements, you cannot use a general all-purpose design. You need something more custom, which is what Paul did so beautifully.
Yeah, really nice. Actually, there is one comment from Olayinka Ashiru. She's asking for if there would be opportunities for research collaborations.
Hit me with an email.
Always open to collaborate.
Happy to play.
Yes, I would suggest to get in contact with them. They are really nice people. I'm sure something will come out. I had another last question. It is the data that you show on plastic genomes and transposable elements. If I remember correctly, this was based on genome scheming data.
Yes.
But I guess you also get such DNA as a bycatch from target capture.
That's a very interesting question.
At some point. So is it enough what you get as bycatch? Probably not to get a full picture of transposable elements, but probably yes to get plastic genomes. I don't know.
I can.
I'm just going to do a
Okay.
molecular methodological comment, and then you can continue. So one of the things that you do when you capture with your baits in the hybridization, there is a step where you use blockers. Okay, those blockers sometimes are proprietary or not, but most often these blockers target organelle or DNA and the ribosome. Okay, so if you do a target capture sequencing reaction with blockers, you're only going to get your single copy, and you're completely going to bias your high copy number portion. When you do the scheming, on top of when you do the HypeSec approach, which is mixing the enriched with a little bit of the unenriched libraries, you're gonna boost again the signal of those organelles, but again, they're gonna be biases. And that said, please, Paul.
Thank you, Lisa, for the introduction. But yeah, I analyzed that quite a bit, so I can give you a good answer. If you want, if you want to work with organelles, HypeSec should be enough. Sometimes target sequencing alone can get something, but it's very residual, and I would not rely on it to do a study because it's too few data. Basically, the blockers work very well. But with HypeSec, you can do the plastic analysis, but for the repetitive element composition analysis, you really have to do scheming alone. Why is that? Because there is, if you do HypeSec, for example, and you take out all the data for the genes, there's still a bycatch when you do target sequencing. There's some things that are still there. And this biases the library, the data set coming from the HypeSec, and gives very different results. I have compared data sets that come from HypeSec and that come from scheming, and the genome composition results are basically not reliable when you do HypeSec. So I wouldn't use that for genome composition. If you want to study a satellite, for example, that is very abundant and you are just interested in that satellite, go ahead. but for whole genome composition.
Perfect. That's a perfect answer. Thanks, Paul and
You're
Lisa.
welcome.
So I think we have no more questions as far as I can see. So with that, I would like to thank you again for your time and for showing us all these interesting methods and applications in real species. Very interesting friends. Thanks a lot, and see you next time.
Thank you.
Yeah, thank you for having us. Thank you.